Good morning to everyone present here, respected dignitaries, esteemed faculty members, and fellow participants. My name is Deepshika Tiwari. I would like to welcome you all to the third day of this workshop themed Drug Discovery and Development. Drug Discovery and Development from Molecule to Medicine with AI, being held at Samrat Vikramaditya Vishwavidyalaya, Ujjain. Now, I would like to welcome all dignitaries for Saraswati Puja. To begin, I kindly invite Dr. Kamlesh Dashora sir to present a bouquet to Honorable Vice Chancellor Dr. Arpan Bharadwaj sir as a token of our warm welcome. Now, I kindly invite Dr. Praveen Khidwarkar sir to present a bouquet to Dr. Shirish Sherlekar sir as a token of our warm welcome. It is my honor to introduce Dr. Shirish Sherlekar sir, a distinguished surgeon and academic leader. Sir hails from Mahu and is an alumnus of MGM Medical College, Indore. With an illustrious career spanning over 4 decades, Sir has contributed immensely in the fields of clinical medicine, drug discovery and development, medical device research, and the application of artificial intelligence and machine learning in healthcare. After years of dedicated clinical practice at institutions such as PT Hinduja Hospital, Mumbai, and Kaiga Nuclear Power Corporation Hospital, Karnataka, he transitioned into the pharmaceutical and biotech industry, working with leading companies such as GSK, Pfizer, and AstraZeneca. Later, Sir played a pioneering role at Tata Consultancy Services, where he headed the Life Sciences and Healthcare Unit in Indore, establishing global drug development and medical device research services. His leadership spanned 18 years at TCS, building a team of over 15,000 professionals across multiple countries. Post retirement, since 2024, Sir continues to serve as an advisor to the biopharm and medical device industry and is deeply passionate about nurturing young talent by teaching and mentoring students in the field of drug and device discovery. Respected Sir, we are truly privileged to have you among us today. Yesterday, we learned about clinical data management, biostatistics, and medical coding. Today, we will explore topics such as pharmacovigilance and drug safety, which plays a vital role in research by ensuring the safety and effectiveness of drugs and vaccines by detecting, assessing, understanding, and preventing adverse reactions and other drug related problems. Let us all welcome Dr. Shirish Sherlekar sir with a big round of applause for today's session.
हेलो
सर, मैं थोड़ा सा बताना चाहता हूँ। आज की जो ऑडियंस है वो थोड़ी फोरेंसिक साइंस से रिलेटेड है और बच्चों ने बहुत ही हमारे यहाँ पे अक्रॉस द कंट्री पूरे बच्चे केरला, उड़ीसा सब जगह से ज़्यादातर बच्चे आउट ऑफ द स्टेट है। तो थोड़ा सा अगर फोरेंसिक साइंस का भी इसका अगर यूटिलिटी आप कर सकते हैं तो बहुत अच्छा रहेगा ताकि बच्चे बेनिफिशियल रहेंगे।
हेलो
गुड मॉर्निंग। सवालों जरूर पूछिएगा। आज के सेशन में जो सबसे ज्यादा सवाल करेगा उसको अवार्ड मिलेगा मेरी तरफ से, ठीक है। जो सबसे ज्यादा सवाल करेगा उसको इनाम मिलेगा और जो चुप बैठा रहेगा उसको सजा मिलेगी। चुप बैठेगा तो आपस में बात नहीं करनी, सर से बात करनी, ये तो अभी शुरू हो जाओगे आपस में बात, ठीक है। क्या हो गया? एक अद्भुत अवसर है जुड़ने का सर से। सर वहां पर बहुत बड़ी भूमिकाओं में है और बड़ी कृपा पूर्वक सर के पास बहुत समय की कमी होती है। अधिकांश समय सर विदेशों में रहते हैं। ऐसे समय में उन्होंने बहुत मूल्यवान समय हम लोगों के लिए दिया और वो समय थोड़ा नहीं, 5 दिन। आप कल्पना कर सकते हैं कि अपने परिवार से दूर, अपनी पत्नी से दूर, अपने बच्चे से दूर कोई आदमी 5 दिन रहने के लिए आया तो वो किसके लिए आया हुआ है। ऐसी ही कल्पना आप क्या हो गया, लाल शर्ट वाले बच्चे सीधे बैठिए। वो आप बच्चों के लिए, अपने बच्चों को छोड़ करके, अपने परिवार को छोड़ करके आए। तो अभी आपका दायित्व यह है कि उनकी बात को ध्यान से सुने और उसमें से क्या आप एक्सट्रैक्ट कर पाते हैं, क्या आप ले पाते हैं, क्या आपके काम का आप निकाल पाते हैं, उस पर आपका ध्यान होना चाहिए, ठीक है। और कल तक, आज ये तीसरा सेशन है। पिछले 2 दिनों में हमको जब सर ने कुछ सेलेक्टेड बच्चों के साथ में सेशन शुरू किया था तब समझ में आया कि चलिए इसका फायदा दूसरे बच्चों को भी पहुंचाएं, ऐसा प्रयास करें। अन्यथा ये सेशन 2 दिन से चल रहा है, तीसरा दिन है। तो ये जो फोरेंसिक के बच्चों को बुलाया है वो इसके लिए बुलाया है कि आपका कुछ बेनिफिट हो। आपको जिसको ऐसा लगता है कि भाई मेरे काम का नहीं है वो कल से ना आए, कोई बात नहीं। आप अपने जहाँ पढ़ रहे थे वहाँ डिपार्टमेंट बैठ के पढ़ाई करते रहे। आज लेकिन आप पूरा ध्यान से सुनेंगे। जिसको रुचि बनी रहती है वो यहाँ पर सुनेगा, अपने डिपार्टमेंट जाके नाम बता देगा कि सर मैं मैडम, मैं आपके अंजलि मैडम को, गणपत सर को या दूसरे फैकल्टी मेंबर्स को बताएंगे कि सर कल से मैं वहाँ कंटिन्यू करना चाहता हूँ। लेकिन यहाँ बैठ करके किसी प्रकार की आपस में बातचीत या इनडिसिप्लिन एक्सेप्टेबल नहीं है, आपको बताना चाहता हूँ। क्लियर है सभी को? अभी भी किसी बच्चे को लगता है कि पता नहीं मेरे को तो मजा नहीं आएगा, तो आप जा सकते हैं उठकर। पहले बीच में अगर उठकर जाएंगे तो फिर बात ठीक नहीं होगी। एनीवन, आपको अगर जबरदस्ती लाया गया है, आपको किसी ने कहा है कि नहीं आज वहीं चलना पड़ेगा, तो मैं आपको बता रहा हूँ यहाँ आपके बैठने की बिल्कुल जरूरत नहीं है। आप जा सकते हैं, आपकी क्लास आपके डिपार्टमेंट में लगेगी। लेकिन बीच में से उठ करके जाएंगे, फिर एक चलता है उसके पीछे सारे जाने लग जाते हैं, ये नहीं चलने वाला है। आपको मैं अभी बता रहा हूँ, कोई भी व्यक्ति जिसको काम है, अभी बैंक आपको जाना है, फॉर्म भरना है, फॉर्म जमा कराना है, जो भी काम है आप जाइए अभी उठ करके, बिल्कुल आने के लिए कोई घंटी नहीं बजेगी, आपको जब आना तब आइए, तब सुनिए आप, कोई बात नहीं, जब से सुनेंगे तब से फायदा मिलेगा। लेकिन छोड़कर जाने से आप एक नेगेटिव प्रभाव दूसरे बच्चों के ऊपर भी डालते हैं। जो ऊहापोह में होता है ना कि मुझे जाना चाहिए कि नहीं, वो भी आपकी प्रेरणा से जाने लग जाता है। तो जिसको जाने का मन है, सुनने में दिलचस्पी कम है, रुचि नहीं है अध्ययन अध्यापन,
Hello. Good morning to everyone present here, respected dignitaries, esteemed faculty members, and fellow participants. My name is Deepshika Tiwari. I would like to welcome you all to the third day of this workshop themed Drug Discovery and Development. Drug Discovery and Development from Molecule to Medicine with AI, being held at Samrat, Vikramaditya Vishwavidyalaya, Ujjain. Now, I would like to welcome all dignitaries for Saraswati Puja.
पूरा भारत है फोरेंसिक डिपार्टमेंट खास करके, तो भारत कैसा है, भारत का युवा कैसा है, ऐसा सर देखेंगे। ठीक है, तो बहुत विश्वास के साथ में आपके प्रति बहुत विश्वास है आप बच्चों पर कि हमारे बच्चे बहुत अच्छे हैं, हमारा नाम ऊंचा करेंगे, इस दृष्टि से सर को बुलाया। आपसे कुछ ले नहीं रहे हो, ठीक है ना, आपको देने के लिए आए और बड़ा त्याग करके आए। मेरे से कोई 5 दिन घर छोड़कर जाने के लिए कहे तो मेरे को बड़ा संशय हो जाएगा। एक आध दिन तो चला जाते हैं, माँ की तबीयत का क्या होगा, पत्नी का ये क्या होगा, उसको बुखार आ जाएगा तो क्या होगा, बच्ची आने वाली है, आदि आदि आदि। तो दिस इज नॉट ए इजी जॉब, बाहर निकलना भी, घर के बाहर निकलना भी कठिन होता है। तो इसका सम्मान करेंगे और सीखेंगे, ठीक है। और श्रेष्ठतम गुरु आपको मिले, आप ये समझ लीजिए, श्रेष्ठतम इस क्षेत्र के जो श्रेष्ठतम वैज्ञानिक हैं वो आपके सामने। अगर आप उसके बाद में भी कुछ लापरवाही बरतते हैं तो फिर आपका दुर्भाग्य है, ऐसा कहना चाहते हैं। पर पूरा विश्वास है, अनुशासन का पालन करेंगे और सीखने की हर चेष्टा को सफल करेंगे। थैंक यू सो मच।
Good morning. I said good morning. How many of you are from forensic? Okay. Is toxicology part of it? Is toxicology taught? पढ़ाते हैं क्या toxicology? हाँ, आगे के सेमेस्टर में किसी ने अभी तक पढ़ी है toxicology? कहाँ यूज होती है टॉक्सिकोलॉजी? हाँ, IDRS. What is IDRS? Sorry, I don't know what is IDRS. Adverse drug reaction. ADRS. Where else? So sorry. Toxicology is part of preclinical. Now, many of you who are from forensic have not attended the past 2 lectures. So, before I come into pharmacovigilance and how toxicology is related to pharmacovigilance, I'll give you a little brief of what clinical trial is and why toxicology is so important. It will also help the students who had come for the last 2 days to understand the connection between toxicology, drugs and human use of drugs. Now, yesterday when we spoke, and this is mainly for the benefit of forensic guys, clinical research or drug discovery and development is a process which spans about 15 to 20 years. It's not a short term thing कि आज आप गए एक disease आ गई और कल आपको मिल गया. Covaxin or Covishield or the messenger RNA vaccines were a bit easier to develop क्योंकि वैक्सीन जो कोवैक्सीन था it was a dead attenuated virus, so it's easier to kill a virus but still retains its potency to develop antibodies. So that development is a bit easier, and because it was in an emergency required, a large amount of requirement for Covaxin, Covishield, the messenger RNA vaccines were not put into place because you had a choice to either allow the patients to die or you had a choice to take a little bit of a risk and start vaccinating population. It's true for all the COVID vaccines across the world, including US and Europe, where this risk was taken, similar to what was taken in India. Now that particular thing came very quickly into the market, but by and large, a drug takes about 15 to 20 years before it can come into the hands of a doctor to prescribe. And even after when it comes, the research still continues for another 2 to 3 years before you can confidently say that now you are in safe hands. After doing all this trial work of 3 phases of animal testing, of toxicology, there are still chances that the drugs can be withdrawn from the market. And in the history of pharmaceutical industry, there are many cases as recent as about an year and a half back, 3 years back, 5 years back, that the drugs were withdrawn from the market because of serious adverse events or deaths. As an example, if I give, was Vioxx, which is known as the COX-2 inhibitor. Then you had Avendia, which was an anti-diabetic compound. And you had certain statins which were withdrawn from the market. So there are many examples which tell us that no medicine is safe, no matter what investigations you do, as many clinical trials you do, no medicines will be safe ever. Anybody who says, including paracetamol, who we take in right, left and center, paracetamol is one of the most dangerous drugs that you can take. How many of you know about that? How many tablets of paracetamol can cause a problem? More than 4 grams. How many tablets it is? It's about 4 grams. 500 milligram का एक tablet आता है या 650 milligram. In Europe and in US, every year about 150,000 मतलब 1,50,000 लोग एडमिट होते हैं because of paracetamol poisoning. 1,50,000 उनकी सबकी liver fail होती है, and once it is damaged you cannot recoup the liver. So no drug is safe. All drugs need to be taken with caution, and all over-the-counter medication should be stopped by people who do not understand medicine, not pharmacy, medicine, which is a far broader area. Now if we come to drugs, now as we know, the drugs has an effect which is desirable and an effect which is non-desirable. Non-desirable can be toxic, non-desirable can be fatal, can lead to serious problems or even withdrawal of the drug from a given patient. All this falls into the domain of what is known as pharmacovigilance or drug safety. As far as toxicology is concerned, it is extremely important because those toxicology studies are done on animals in the preclinical phase, and they are done on various animals at various doses to understand not only toxicology, because toxicology consists of a particular dose, what is given, and what is causing harm predominantly to the kidney and the liver and the heart. These are the 3 systems where they predominantly look for toxic effects of a drug. And beyond that, they look for toxic effects on whether it is causing a congenital abnormality in the newborn, and so it is given in pregnancy in rats and guinea pigs and all, to understand its cancerous effects and its effect on how it can cause congenital abnormalities. So these are certain uses of toxicology which occur in the earlier stages of a drug development, and then those parameters are used to decide a dose in humans. So always the dose in animals is different than the dose in humans. No matter how similar the animals are to humans, guinea pigs and all are common and similar, there is still a huge amount of difference between animals and between humans. And within humans also, there is a difference between young population, that is pediatric, between pregnant women and geriatric population, and the population between the age of say 25 to about 60. So there is a huge difference in that, and hence the field of toxicology is the base on which pharmacovigilance stands or drug safety stands. So if you don't have toxicology data, you cannot move into further trials in humans. And hence, toxicology is something which is extremely important. Now you are all from forensic medicine, many of you. I have one question. Can apple be toxic? How? No, can apple per se be toxic if eaten? How? No. Can it be toxic? Can it kill you? Anyone? How many? Agatha Christie padha hai kya? Apple seeds are extremely poisonous. A handful of apple seeds can kill you. And hence, if you see, animals don't eat apples. They very rarely eat apples. They don't. So those are certain ways of identifying toxicity of compounds as well. And hence, further it goes to pharmacovigilance, which is our topic today. Next slide. Next one. Now, what is drug safety? There are a couple of words here which are extremely important to understand drug safety of pharmacovigilance. Pharmacovigilance, it is a science. It's a science of activities which deal with detection. Detection is identification of an adverse event. An adverse event means which I do not want. And as an example, I take aspirin. If I develop gastrointestinal discomfort, acidity, regurgitation, that is not what I wanted. I wanted just a pain-killing effect of aspirin. Now, if you go to a doctor after taking aspirin with this complaint, the doctor has to ask you a history, and that is how he will detect whether an adverse event has occurred because of the drug, or he will at least suspect it. Then he has to assess it. You go to a doctor, it is not necessary that aspirin causes discomfort and abdominal distension and acidity in every patient. It may not cause in many patients also. So he has to assess that whether that nausea, vomiting, abdominal discomfort is because of taking aspirin or it is because of some other reason which a person has. Simple thing, a case of gastric ulcers or duodenal ulcers. Is it occurring because of that? So he has to assess that. And after you assess and detect it, you have to understand how to prevent it in the future. The use in the pharmaceutical industry, both of toxicology and pharmacovigilance, is that as they manufacture and create new drugs, they use this data and try to minimize these effects in the next drug for the same disease that they develop. Now, you all have heard of propranolol, then etanolol. These are those LOL series of drugs which lead to, which were used in hypertension. Now each one, you had cimetidine, you have ranitidine, then you have omeprazole in the same different class compounds but used for the same disease. Cimetidine had a problem of males developing breast, which is known as gynecomastia. That particular adverse event occurred rarely, but that was taken care of in ranitidine by changing in the molecular structure. Now ranitidine has also a problem to a lesser extent of gynecomastia than cimetidine. So it was an improvement. Now you have omeprazole, which is a different mechanism of action, but omeprazole, if you take chronically, it can lead to gastric hypertrophy. So that is how you detect, you assess, you understand, and then the next molecule you develop, you try to minimize the side effect of it or adverse event. And this occurs in medicines, vaccines, devices, anything which is used on a human being for treatment, prognosis or diagnosis. This particular thing comes into play. There is pharmacovigilance for devices also. Very few people pay attention to that problem, but there is a pharmacovigilance for devices, medical devices which are used in humans. So you have heard of robotic surgery? आजकल बहुत एडवर्टाइजमेंट्स चल रहा है, रोबोट से सर्जरी होता है. वो रोबो इज नॉट कि ओटी में छोड़ दिया रोबो को, रोबो गया चढ़ के उसने ऑपरेट कर दिया. वो पीछे एक सर्जन बैठा रहता है जो रोबो को चलाता है. So if something goes wrong, and robo is electronic, so if something goes wrong in the robo or the commands do not reach, the robo can spoil the operation and create a problem. The most famous robo was for robotic surgery was Da Vinci. Da Vinci नाम का एक रोबो है जो अमेरिका में किसी कंपनी ने बनाया था. That Da Vinci caused a huge amount of trauma to people on which it was used for surgery. That was an adverse event of that Da Vinci robo. It has been improved now, but there also adverse events occur and they need to be reported, analyzed and used. All medicines and vaccines undergo, in clinical trials, we undergo clinical trials through 3 phases and we go animal studies. So we do a huge amount of testing, but very unfortunately, when we do testing in a scientific setting, this clinical trial, the trial and the drug is controlled, the population to whom it is given is controlled, and as a result of which you do not have variations in which you give. When the drug is approved and it comes into the market, to which patient it will be given as per the clinical trial or as per a general is not known. So tomorrow, if you have a patient of diabetes, he has hypertension also, he has cardiac problem also, he has kidney problem also, and if you give him a drug, the way the drug will act or react in the patient will be different than what it would have shown in a clinical trial. And hence it is important that after the drug comes into the market, the pharmacovigilance continues. In clinical trials, the maximum total patients that you would have used is about 5,000 or about 6,000 patients. But the moment it comes to the market, within 30 minutes of it coming to the market, you would have had about 1 million prescriptions across the world. So करीब 10 लाख लोगों ने वो अगर टैबलेट एक झटके में ली, तो won't you think that some other adverse events will crop up? I'll give you an example of that. How many of you know about cetirizine? Does cetirizine cause sedation? Does cetirizine cause sedation? Well, it is not supposed to cross the blood-brain barrier, no? So how does it cause sedation? Now these are typical marketing gimmicks which they come up. If the company was so confident that cetirizine or levocetirizine or fexofenadine and all does not cause sedation, then in the precautions and warning sections of the prescribing information, they should not have written that please drive carefully if you are taking cetirizine. Why do you want to write that? So these are certain, and it is a so-called second generation anti-allergic. What is second generation anti-allergic? These are not pharmaceutical terms. These are not medical terms. These are marketing terms. What is the first generation, second generation, third generation cephalosporin? Marketing terms, not medical. कभी किसी ने pharmacology में पढ़ा है? आजकल books में आ गया है, unfortunately. But there is no classification like first, second. It's only gram positive, gram negative, or a broad spectrum antibiotic. There is no generation. So these are all things which come up because of marketing. And though we test it vigorously during clinical trials, the sample sizes is very low. Whereas when it comes into the market, the number of people taking medicine is suddenly high, one. Second, to whom it is prescribed is not controlled. It's up to the doctor. So he may prescribe it to anybody he thinks is correct or not correct. The third is the variation in the genetic material of different countries also has an impact. A diet has an impact. And on top of it, in countries in Asia and Africa and Latin America, Ayurvedic medicine, Unani medicine, Siddha medicine, African medicine, Egyptian medicine, which is based on various types of therapies, can also interfere into the drug. And I am talking of Ayurveda in its true sense. I am not talking of Ayurveda which is sold over the counter. Like Himalaya and Dabur and everything now is Ayurvedic. I am not talking of there, because Ayurveda is an extremely scientific branch, as similar as allopathy. Only thing is the way it treats patient is different and effective. But today Ayurveda has been reduced to घर में जो भी कुछ तुम्हारे घर में मिलता है सब आयुर्वेदा है और मैंने बोल दिया तो आयुर्वेदा. That is not Ayurveda, because Ayurvedic medicines also have side effects written in Charak, written in Shushruta. And those are the two most sacred texts of Ayurveda. Also in Vagbhat it is written. If any one of you can read, go to the internet. All the 3 books are available on the internet. So even Ayurvedic preparations has side effects. To give you an example, Trifala Churna. And everybody knows, made up of 3 things. If you take more, it will lead to diarrhea. It's supposed to relieve constipation, but if you take more, it will lead to diarrhea. And Trifala, you have to take it in a particular way, not chammach muh mein dal ke pee liya pani and ho gaya kaam. That's not the way to take Trifala. But nobody tells you that, because marketing takes over. And today you don't have, otherwise previously Ayurveda had a pharmacopoeia, which has gone missing. Ayurveda has dose schedules missing. So don't think that Ayurveda is something which is kuch bhi kha lo aur hum safe hai. You eat haldi of 3 teaspoons a day directly and you see next day what happens to you. Haldi to hum roz khate hain. Khao jara haldi 3 chammach roz. और फिर देखो क्या हालत होती. So pharmacovigilance is required everywhere as in everything. Next one. So the word pharmacovigilance comes from Greek. Pharmakon is drug or medicine. Vigilia is to keep a watch on the medicine which you are giving it to the patient, and that is what means pharmacovigilance. Next one. Now why did pharmacovigilance come up? How many of the pharmacy students have seen this photograph? What is it regarding? Called? Congenital abnormality of? There was a drug called thalidomide. Thalidomide was discovered in Germany in the early 40s. And as I said in my previous lecture, at that time, clinical trials or anything was not existing. A chemical company used to make it. वो कंपनी कंपाउंड ले जाती थी डॉक्टर के पास और बोलती थी इसको यूज करके देखो हमारे हिसाब से एबीसी डिजीज में ये काम आएगा. नो एनिमल स्टडीज, नो ह्यूमन ट्रायल्स, नथिंग. एंड द डॉक्टर डोज क्या है तो वो कुछ भी बता देता था केमिकल कंपनी वाला, बिकॉज देयर वाज नो फार्मास्यूटिकल कंपनी एग्जिस्टिंग. और इस चक्कर में थैलिडोमाइड वाज टू बी गिवन नॉट फॉर nausea and vomiting. Thalidomide was to be given for something completely different. However, as the tablets became available, the name of the company which manufactured it was called Grunenthal. That company still exists in Germany, and now it is a completely pharmaceutical company called Grunenthal. Now that particular company put it into the market in 1950. No research was done on the LD50 level. You had an LD50 dose for everything. No studies done. It was approved in Germany in 56 as an OTC. Till that point of time it was in prescription. Soon about 14 companies started manufacturing it, because in 50s there were no patents. So 14 companies manufactured and put it in 59 countries except the US. US for some reason did not allow thalidomide to be put into that. Now it was started using for nausea vomiting. Initially it was like a sedative or a tranquilizer. And when it started, they started taking for nausea vomiting, the incidence of intake by pregnant women increased exponentially. And as a result, they had these kind of children born, that is children with no legs or very short legs and no arms or very short arms, a congenital abnormality which is known as phocomelia. So like that, 13 reports came in 61, and by the time it was withdrawn, about 10,000 children were born with phocomelia. And the reason being, there was no pharmacovigilance at that point of time. And hence this occurred. I can give you a very recent example of even with the existing pharmacovigilance, there were cases of serious adverse events and deaths when people were taking certain antipsychotic compounds. Antipsychotic compounds, one of them was the serotonin inhibitor, uptake inhibitor. So that particular compound was marketed for adults. The said company did a trial, a marketing trial, so no approvals were required. So did a marketing trial in adolescent population. Adolescent population is between 12 and 18 years old. They did a study there and they proved by the trial that it is useful in adolescent psychiatric illness or anxiety or depression. Medically, anyone below 18 years does not have depression, cannot develop depression. The disease of depression is not reported anywhere in the world in the pediatric population. Nonetheless, the trial was done, it was given, and there were thousands of suicides of these children. But nobody took care of it. The clinical paper was published in the Journal of Psychiatry in US. That paper was circulated amongst the psychiatrists, like any psychiatrist had got that paper from the company, and all of them started prescribing it. As a result of which, number of suicidal deaths in adolescent population started increasing. It continued for about 8 or 10 years, and then a psychiatrist from UK called David Haley started researching it and asking for details. So the first paper was published in the early 80s or something in the Journal of Psychiatry. And after a long legal battle with the said company, pharmaceutical company, he got the source data from the company. So a group of psychiatrists analyzed it and they proved that it is not useful in the pediatric population, and given to them, it leads to suicidal tendencies. Now, yesterday I spoke to you about coding and how coding is critical. Now, if you see, and both the papers are today now on the website. If you want, you can type trial 329. The trial is known as trial 329. You will get both the papers, an old paper and a new paper. New paper is done by David Haley. Exactly using the same data, exactly opposite results calculated. Now, why it occurred? There were many reasons for it. One of the important reasons was coding, medical coding. Now, as we saw yesterday, if you want to code, you need to code it correctly. And this code keeps evolving. Now, when the pediatric patient came to the doctor, and the doctor took a history on the trial, he said, "How are you feeling?" नहीं अच्छा नहीं लग रहा है मेरे को, I feel like अंधेरे में बैठने की इच्छा हो रही है. Another patient said that, "No, I want to jump off the window." Third patient said, "No, I feel like not living." Now all these symptoms or talk which he is doing means that the child has a suicidal thought. आत्महत्या करने की उस बच्चे में इच्छा जागृत हो रही है. But how was it coded medically? इमोशनली लेबाइल, इसका इंग्लिश में मतलब इतना ही है कि वो लड़का थोड़ा अपसेट है इमोशनली. How different is emotionally labile or being upset with having a suicidal tendency? And that is how a wrong coding was done, which resulted into doctors feeling, oh, it's not a major problem. Emotionally labile, तो anyway the child is depressed, so होना तो है, उसको anxiety तो रहेगी, so what is the problem? So be very careful, and that is why coding, as I said yesterday repeatedly, is one of the most critical steps in clinical research. You do a wrong coding and you will have a huge number of dead bodies next to you. So be careful about it. So this is why pharmacovigilance actually began. Go to the next one. Now why is PV needed? As I said, in clinical trials the sample size is very small. And when it comes into the market, billions of people use it. So new reactions can be exposed. You can have small, less incidence of a reaction to increase in its number. Example is cetirizine. Cetirizine is not supposed to cause as per the prescribing information in clinical trial. But now if you see, there is a huge amount of sedation with cetirizine, fexofenadine, levocetirizine, everything. So those are the ways you figured it out by doing pharmacovigilance. Second, the population in which you use changes. So a trial may be done in US and Europe, but it is being sold in India or in China or in Latin America. Population is different, diet is different, genetic composition of the person is different, so it can cause it. As I said, no drug is safe. And as I said before, adverse drug reaction is the 4th to 6th, depending upon who did the trial, 4th to 6th largest cause of death in the world. And this is after only 10% of adverse events are reported to the government. So if hundreds have occurred, 100 adverse events have occurred, only 10 get reported. You must have also gone to a doctor, right? Every one of you or your relatives. Does the doctor anytime ask you, गोली लेने के बाद कुछ तकलीफ हो रही है? और अगर डॉक्टर ने कभी गलती से पूछ लिया तो पेशेंट क्या कहता है? सर दवाई तो ली है, अब तकलीफ तो होगी. But in that 2 sentences, what you are denying people is the scientific information of that adverse event, which will lead to more safer drug. I am using the word safer, not safe. And hence, anything happening to you as an adverse event, known or unknown, it is your responsibility as a patient to tell the doctor. It is doctor's responsibility to ask you. And then it is your and doctor's responsibility to report that adverse event to the company. And all companies have a website on which you can report it. Drug controller CDSCO also has a website where adverse events can be reported. WHO also has a website. US FDA has a website. UK MHRA has a website. And you sitting in India can put the adverse event there. But there is in general a hatred amongst the patients as well as the doctors, the pharmacists, the nurses to report an adverse event. And hence we still have drugs which have not become safer than what they were in the past. We still are taking dangerous medicines. It helps to use rational use and helps doctor in prescribing. Both the statements are negated because nobody does that. Next one. Another reasons why PV is needed is drug production and formulation is different. The API may be the same, but API can be produced by 2 different companies under 2 different quality standards. Second, in the formulation, you also have excipients. Excipients may be inert, but they have a potential to cause an adverse event. And hence, many times when you take a drug here, you may have no adverse event. If you go to UK and take a drug, you may have an adverse event for the same drug which you have been taking for long. So hence, pharmacovigilance is needed. Dose availability and indications. For what it is used varies from country to country as per approval. Dose changes from country to country. And more importantly, like paracetamol, patients take it irrespective of the dose. If you have a cold and cough, and how does poisoning occur with paracetamol? First of all, you have cold, cough and fever, so you will take a paracetamol because you have fever, though it should not be taken. So you take a paracetamol. Then अभी खांसी भी हो रही है, तो अभी क्या, तो कोई कफ सिरप ले लो. You don't realize that even in the cough syrup there is paracetamol. फिर अरे सर्दी भी हो रही है, तो चलो एक और टैबलेट खा लो, उसमें एंटी एलर्जिक है. Now you don't realize that with that anti allergic is also a paracetamol. तो तुमने एक डोज में 150 ग्राम पैरासीटामोल ले लिया. अब दिन में तुम 4 डोज लोगे तो क्या होगा, वो जो होना है वो होगा. And hence it is to be careful. It is not the tablet, as he said rightly, it is not but the quantity of the drug which is to be calculated, taking into consideration all the medicines that you are taking. Quality, as we discussed yesterday, of drugs vary, whether it started from 105% purity or 90% purity. So that can also lead to a problem. The classic example is ranitidine, which has been withdrawn world over except in India. Ranitidine during the manufacturing process develops a cancerous compound within it, and when it is on the shelf, because of the temperature and humidity, it develops a chemical called NDMA. NDMA. Look out for what is NDMA. I am not going to give you the full form. That NDMA is a cancer causing agent, and hence the drug has been withdrawn world over. In India, unfortunately, it still exists. It helps analyze the risk. If I know what is the incidence of this adverse event, it will help me reducing my prescription only to those patients who are needing it very definitively. Helps advance safety and benefits, ethical concern, and doctors are then more confident of prescribing, and you will be more confident of taking the drug. Next one. Now, when we talk about PV or drug discovery and toxicology, people usually think that the first step, which is single case report, ICSR is the single case report which comes from the market or the doctor, pharmacist, nurse or anyone. Understanding of how to process it means you are masters of pharmacovigilance. But that particular ICSR is the first step of pharmacovigilance. It begins there, it doesn't end there. But even if you go to the pharma companies today, majority of the people you will see saying, oh, I am an expert in pharmacovigilance. I still don't call myself an expert, because in medicine you cannot be an expert at all. You will always be a learner. And if you don't learn, you're doomed. And why I am saying is, I also, like a student of yours in medicine, was taught by a very famous cardiologist of Indore called Dr. S.K. Mukherjee. Dr. S.K. Mukherjee was the cardiologist to the President of India at that point of time. I'm talking of 1985. When we came into third year, Dr. Mukherjee used to teach only third year students. Only third year students, nobody else, and in clinics. At that time he was about 85. So he used to come, sit on a chair. We all used to stand around the patient. And his first question on the first day of the clinic to every batch was, how many of you think that you will be able to pass MBBS? All students, sir, yes, 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 yes. So he used to be having a smile on his face and say, तुमको मालूम है कि MBBS की डिग्री दी जाती है, ली नहीं जाती. बोले मेरे को भी किसी ने दी है, मैंने ली नहीं है. मैं कितना ही पढ़ लूं, no matter what books I read, no matter how many years I read, if I am asked one question and if I can't reply to that question adequately, the patient is gone. So he says, are you then good doctor? So he says, we are all good or bad doctors where degree somebody senior of yours has given to you. So never get into this impression that I am a very big doctor. Always think you are the most useless of it and you are trying to do your best in the given circumstances. That's all. So that is why it is said that in medicine, continue to learn till you stop practice or till you die. The day you stop, your patients are in danger. So that is something which is very important. So in the PV, the total life cycle of a drug is that you get a single case report form, you do a medical review of it to say whether the adverse event which is or because of something else, that is what is medical review. Once you have on an yearly basis a collection of single cases, you write what is known as an aggregate report, which is a safety report submitted to the regulatory authorities that in 6 months, in a year, various timelines for various drugs are there and maturity of the drug is there. तो वो कहते हैं कि 6 महीने में या साल भर में मेरे को उस साल में कितना एडवर्स इवेंट्स हुआ है उसका कलेक्टिव एनालिसिस चाहिए. So that you give it to them, which is in the form of aggregate report. Then there is something which is known as the risk management plans. That as you collect the data, you will find that, oh, the risk has increased or decreased or remains the same. And if it has increased, what is to be done is the most important question, and that is taken into consideration in risk management plans. These are also submitted to the regulatory authorities. Another thing is called signal detection. I'll take each one of them in a little detail later. Then you have a signal confirmation, and the last part which occurs is label. So why you do drug safety is to ensure that the label, the pack insert, the prescribing information which you have changes depending upon what safety data you have generated. And hence, the process which begins at receipt of the case, it ends whether the label has changed or it has not changed. So that is the life cycle of pharmacovigilance. So if you are going into in some of these roles are reserved for doctors because it requires a medical degree, but you can also do signal detection, but confirmation you cannot do because you are not doctors. It will be done by a physician. Similarly, medical review, you can do certain parts of medical review, but the final signature will be done by a physician. So it is not that a toxicology or a pharmacovigilance student cannot do this. They can, but there are limits to what they can do. Toxicology students, the predominant use of them is in preclinical studies, where the animal toxicology reports are submitted to the regulatory authorities, and that work is predominantly done by either toxicologist or the forensic medicine and toxicology, or it is done by veterinary doctors, because they are accustomed to understanding how the drug behaves in an animal. So those are the two branches which go into that field very common. Some of them who are interested in human pharmacovigilance, they come here also. Next one. So PV is, anybody wanting a break I can give, otherwise I can continue for 6-7 hours also. So anybody wanting a break? Now the PV is a teamwork. One single person cannot do drug safety or pharmacovigilance. The first person is the doctor. Patient is obviously before. He complains or doctor understands complaint. So doctor first knows that patient is having a problem or is having an adverse event. Another person who is responsible is the nurse, because nurse are in closer contact with the patients than the doctors. So nurse, if he sees it, she can also report the case. A doctor can report, a patient can report, patient's relatives can report. The third person who is responsible is the pharmacist, because he is the one who dispenses the medication. The pharmacist's unfortunate role in India is very different than a pharmacist's role in Europe or in US. It's very different. There they have the authority to also prescribe and give certain medicines, not all. And they can also report adverse events. Obviously the patient, and then the last is pharma manufacturing. Now understand, adverse events occurs also because of problems in manufacturing. I'll give you an example which I have faced. One of our relatives was having asthma. So the drug prescribed was Symbicort. Symbicort, the drug is old. It has Formoterol and a steroid, budesonide. But the way it is given is through a device which is known as disc healer. Now when the patient started taking it, the asthma didn't improve, and we kept increasing the dose. Over a time I suddenly realized that probably the problem lies in the disc healer, not the drug. We changed the disc healer to normal inhalers, and the problem vanished. So I complained to AstraZeneca, and not here because India nobody bothered about it. I fortunately knew the head of drug safety in global pharma, AstraZeneca. So I sent that disc healer to Sweden, from where the factory is and from where it comes. I could do all this because I knew the whole jing bang of AstraZeneca. Otherwise, it is very difficult. And they told to me one-on-one orally that yes, the descaler has a problem. So I sent an official adverse event to them. The answer came to me that thanks for it, but the incidence is very low. So this is to protect the drug. So there is nothing wrong in the drug. The wrong was in the device. So those kinds of problem occurs, and hence manufacturing companies come into play. Another which I gave you an example some time back was ranitidine. NDMA develops in ranitidine during the process of manufacturing. Another example I can give you, which is very common in cough syrups, is diethyl glycol. Instead of diethyl glycol, to make it cheap, people have put glycerol into it, resulting into deaths or blindness and so on and so forth. So hence, pharma manufacturing becomes extremely crucial in terms of drug safety. Next. Now these are three terminologies which you will get on a regular basis in pharmacovigilance. Any difference? No, don't read that and tell me that I will also read and I know what I have written there. So do you think there is any difference between the three? When you take a drug and something happens to you which is unintended, I didn't want that to occur, but it occurred. And it occurs at doses normally used in man or humans. That is a side effect. It may be good, bad or ugly. I am not defining that. So anything which occurs which I did not intend to occur when I have given it in the right dose, that is a side effect. Now an adverse event is also a side effect or an untoward occurrence or an unintended occurrence in a patient or clinical trial when it does not necessarily have a causal relationship. What does a causal relationship mean? That if an adverse event occurs, I have to prove as a doctor that the cause is because of the drug. So if I take aspirin and if I develop gastric regurgitation, I'll have to confirm whether it is because of aspirin, because of any other drug which he is taking, or because the person has gastric ulceration or duodenal ulceration, or he has a problem of as simple as bad digestion. I have to differentiate, and that establishment is what is known as the causal relationship between the drug and the adverse event. An adverse drug reaction is a response to the drug which is noxious. Noxious means very bad. I just don't want it. The patient is unable to carry his day-to-day life because of that reaction. It's unintended. At normal doses it occur and there is a causal relationship. You can definitely say it has occurred because of the drug. So the difference between adverse event and adverse drug reaction is, in an event, you have no causal relationship. In a reaction, you do have a causal relationship. So when you analyze an adverse event, when a medical evaluator analyzes the event, he evaluates it for this. Because if it is a reaction, then he will consider it for further evaluation. If it is an event, he will just log it and say causal relationship not maintained. That is also used, but for different purposes. Now, as adverse effect and side effect. Side effect can be very severe or can be very minor. If I give you an example, minoxidil was a drug which was developed for hypertension. It developed a side effect which was growth, hair growth. खास प्रॉब्लम तो मतलब आदमी जिंदा है, उससे मरता वरता नहीं है आदमी. So that can be called as a side effect. Later on it was used as an indication for that. An adverse drug reaction can be normal or can be severe. Take for example aspirin, if you take, you develop nausea, vomiting and regurgitation. If you take ampicillin, amoxicillin, you have severe diarrhea at times. Many, not all, some have, some don't have. If you take sorbitrate, anti-anginal, if it goes a little more, you start having a headache. So that is a side effect of it. Because hota hai, band kar do, thuk do, it will settle down. So that is how it is different. So pharmacology, pharmacovigilance and pharmacology, you have to concentrate on adverse event and adverse drug reaction, not on the side effect. Because when you report, you don't report it as a side effect. You report it either as an event or as a reaction. Okay. Next one. Another type of an adverse reaction is medication errors. Example, similar brand names. Chemist makes a mistake in giving you. You also don't read. Doctor also doesn't bother. And you take a medicine. It is some other compound which you are not supposed to take. That is called a medication error. Or instead of one tablet, आपको ही बहुत चिंता हो गई तो आपने एक की जगह 2 गोली खा ली. So that is a medication error, dosage error. So all this occurs and they have to be also reported. Or you have taken a medication and you did not find any effect is also an adverse event. So adverse event is not only when something bad occurs, adverse event also occurs when nothing occurs. That is, the intended action has not taken place. That also is an adverse event, and hence you need to be careful. Mistakes in prescribing, dispensing. Now dispensing today is not in the true sense of dispensing, because in older times, pharmacists used to be actually compounding the drug in the doctor's premises. Today that doesn't occur. But compounding occurs in a pharmaceutical company. Now there, as I said, a common is diethyl glycol causing a problem in cough syrups. Very, very common because of contamination of glycerol. It's a very common thing. And those kinds of things fall under the medication errors and need to be prevented. Next one. Now, adverse events can be normal adverse event. By the term normal, I mean that they do not cause major problems to you. You are not hospitalized. You can bear with the adverse event, or at the most, if it is unbearable, you can stop and start some other medication. So that is an event which is called as a normal event in colloquial terms, or it is just called an adverse event. Then a reaction can be serious or non-serious. A serious adverse reaction, that is SAE, serious adverse event, or SAR, serious adverse reaction, is life-threatening, मरने की संभावना है. You get hospitalized because of that. That is hospitalization initial. So if you are already in the hospital, and if the hospitalization days increase because of the drug, that is a serious adverse event. It is fatal. It leads to death. It leads to disability. आपने कुछ दवाई लिया, it leads to gangrene. Amputation has to take place. That is a disability. Or if you take, you become blind or you become half blind. Then those are disabilities. Those are serious adverse events. It leads to congenital abnormality. Example which I took is of thalidomide. Nowadays it has gone down dramatically because you don't use it in pregnancy, or it has led to cancer. There are many medications, like if I say ranitidine, if you continue to take ranitidine for a long time, because of that NDMA, it can lead to cancer, some other form of cancer. So these are what are known as, if these criteria are there in any reaction which comes to you or reported to you, then it is considered serious. And these serious adverse events are to be analyzed and submitted to regulatory authorities within 7 days of receipt of by the company. Volume high, not you stand, volume high. I said. Forensic scientist will come into play if the dose which is taken by the patient, either prescribed or unprescribed, has led to that. Or it will occur if the pharmaceutical manufacturing has a problem. But that is initially identified by the pharmacist, the doctor, the nurse, and then if required, the factory will call the toxicologist and do it and ask him to analyze it for toxicology. Then he can do animal studies and find it out. But in terms of analysis of that data, you will analyze it as a toxicologist, hand it over to doctor, and he will analyze it from a human perspective. And that is how the further action will take place. Next one. Now in serious adverse event, there is another terminology called suspected adverse reaction. Now this suspected adverse reaction are the ones which are the most crucial ones. So the first one is called serious as the name indicates, it's usually called as SUSAR, that is suspected, unexpected, serious adverse reaction. So when you are doing clinical trials, suddenly a serious adverse event comes up which you have not seen till that point of time. So you suddenly start suspecting it. You didn't expect it, so it is called unexpected. And then it is a serious adverse reaction. SUSARs also occur after the drug is marketed. You can suddenly get deaths in a population who is taking a drug that is an unexpected and a serious adverse effect and needs instant action. And then there are serious but expected adverse reaction. Simple example if I give you, if you take an oral hypoglycemic agent, a serious adverse event would be hypoglycemia. But hypoglycemia is known. If I don't eat and still take a tablet, if I instead of forgetting, I forgot that I took a tablet and take the second tablet, all this can lead to the serious adverse event called hypoglycemia or low blood sugar, but it is known. So we tell all the diabetic patients, agar wo oral hypoglycemic or insulin pe ho, so please keep a sugar packet in your pocket. Put a chit in your pocket that you are diabetic, contact number so and so, blah, blah, blah. So these are all precautions because a serious adverse event which is expected can occur at any point of time in that diabetic, and these are precautionary measures. Next one. Now when a case comes to you, and today in the situation of social media, a lot of people write false information. About 90 to 99% of information on social media is bakwas. Similar is the case of adverse reaction. Whether it is spiritual, medical, road congestion. I have got on social media photographs of Bombay in rains. I am standing at that place and there is no flooding. But photographs are there. Because that would have occurred some 5 years back. So social media is broadly used today by patients because they are not happy with certain things, not necessarily that they have had adverse events. Similarly, many of the patients write directly to the companies and say that I had an adverse event, all built up story, and say that I had an adverse event. Now, how do you figure out whether it is true? No, you can't do it 100%, but there are certain ways. First is identifiable patient. Can the patient be identified? Not his name, I am not interested, but can I identify that this patient took medicine from this or this doctor prescribed it and it is a valid patient? It is not somebody Tom, Dick and Harry who's game. So you need to have a valid identifiable patient. And there are mechanisms to do it in the software on which you process the case. Is there an identifiable reporter? So is it a patient reporting? Is it a doctor reporting? Is it a nurse, pharmacist? Who is reporting? Or a patient's relative is reporting? Who is reporting it? Is there genuinely an adverse event in that? Because if there is an adverse event, by and large, patient, doctor, pharmacist, nurse writes it very clearly as to what was the symptom or the sign or the lab parameter. They don't write a general comment on what has happened to them. And second, can you medically suspect it to the drug? So these are 5 criterias which, as the case comes to you, these are the first 5 criterias that you look into the written data and accept or reject the case. Now, if any of this criteria is not there, you need to contact that person who has reported. Now, in a social media, I can't even know who has reported because there are a large number of false addresses there. So I can't do it. So if any one of it is not there, I just discard the case. But I don't want to generate junk data. Data also has to be good and neat and identifiable and analyzable. So these are the 5 important parameters on the which I initially see whether the case can be processed or the case can be kept in abeyance. Next one. Now there are 2 types of safety cases which come, or 2 types of ICSRs. One is unsolicited. As the word indicates in English, unsolicited means कि pharma company ने पूछा नहीं किसी से कि तुमको कुछ हो रहा है कि नहीं हो रहा. So hence, such unsolicited reports come from patients, doctors, pharmacists and nurses. And second is solicited reports. Now what are solicited? That complaints have come to the pharma company, they want to independently see whether it is genuinely occurring or something. So what they will do, they will send their representatives which are describing their medicines and ask each of the doctor, "Okay, please tell me how many adverse events have you seen and can you give me the details of it?" And hence they are asked for and hence they are solicited. They can be seen in clinical trials, in reports, in patient surveys, and something which is known as patient access programs, which are very common now in India also. When the cost of the drug is very high, the pharma company usually subsidizes the drug. So you take 2 tablets, they will say, "I will give you 1 tablet free," or you take 1 strip, I will give you 25% discount. When they do this, what they expect from you is that if you have an adverse event, you report to them. So they are soliciting, they are asking you to report, and that is the difference. There is no difference in the way they are processed, but it is just the differentiation of whether it is a solicited report or is an unsolicited report. Next one. Now, how do you manage? There are certain softwares which are available today which manage adverse reporting. The most common is Eris. The software's name is Eris. The other one is known as Argus. These are the two. One is from Oracle and the other is from the company called Eris. Both of them are US based. Now, besides this, many companies world over have developed their own software. So like TCS has developed a software, Cognizant would have developed, Accenture would have developed. So they use it for their own purposes when they are doing the safety services, or they hand it over to the pharma company from where they get it. So like that there are many, but Eris and Argus are the two most common adverse event databases or softwares, as they say, which are being used. Now the first step which occurs is receipt. Now adverse event comes in the form of emails, faxes. Many people have call centers, offices have call centers, so people call and complain about it. So there are various ways by which, and now an addition has occurred is that all pharma companies have to scan the social media. So there are a large number of robots which they use, robotic automations which they use, which scan the social media and say that these are the adverse events which are reported. It goes through the same cycle. So that is the receipt. There are call centers in different countries. There are call centers collectively at one place. So various ways are there. Once the case is received and you have validated the case that yes, we need to process it further, you need to see whether it is a duplicate case. There is a habit of patients and doctors to report the same case multiple times. And if it occurs multiple times and if you start analyzing each case and adding the same reaction, you will say instead of 1 case, there have been 15 cases. So there are ways to check whether it is duplicate or not. If it is duplicate, it is not processed. The original one is only processed. Then you log the case. So once you have done this, you create a case ID. Because if you are doing an analysis tomorrow, you require a case ID, and that is called the logging of the case. Once you have done these 3 steps, you have to identify whether the case is serious, non-serious, fatal, life-threatening, etc. Because if the case is non-serious, you can report to the regulatory authorities in 15 to 30 days time. If it is serious, you have to report it within 7 days of receipt. And if you don't report it, there can be heavy fines on you. Now whether it is raining, whether there is a thunderstorm, whether there is a snowstorm, whether there is a riot, there is a war or anything going on, you cannot stop. This is regulators' requirement. Okay, if you can't do it because you are bombed, then you won't, but then they will still ask you why you didn't, and you need to give an explanation to that. So serious adverse event, SUSARs, are something within 7 days you have to do the entire processing and give it back to them. So that is what is known as triaging, that you differentiate it as simple case, complex case, serious case, non-serious. That is called triaging, because then you say, okay, this is non-serious, I can do it in 15 days, let me finish this. And remember, when cases come to you every day, like the operations which we ran in TCS, so the same thing is in Accenture and all. In one day, you can receive about 4,000 to 5,000 cases in a day, of which about 15 to 20% are serious. And this comes every day, irrespective of Saturday, Sunday and holidays. So you need to, that operations need to be very agile, they need to be very quick, they need to be very fast and managed properly. So after you do the triaging, you generate a report or you do a data entry. Now as I said, now data entry has stopped because capture is electronic. But still in some countries in the world, paper formats are generated and you get a fax or you get a PDF copy. Once you do this, you do the assessment of the case because the data entry is already done. Assessment means you code. Like in data management, you did medical coding. Here also you will have to do similar kind of a coding. So once you do this assessment, you do a quality review. The case is reviewed, whether the data entry was correct, whether there is any discrepancy in the data, all that has been seen, and the doctor will actually say whether there is a causality linked to the drug or there is no causal relationship. You will validate all this and ultimately the case is closed and a report is generated to be sent to submission. And this is more important. See, non-serious, you can report it in 90 days. Serious in 50 days, SUSAs and life-threatening in 7 days' time. These are legal responsibilities of a pharmaceutical company or a biopharma company or a device company. You cannot escape this, and majority of the audit findings which come as warnings and severe are because of non-reporting in time. So be very careful about. So this is a simple diagram of how the case moves and what the role of people. So when you begin your career in pharmacovigilance, you will begin as a person who is receiving the case. And then you will climb up the ladder and then do submissions. I'm giving it simplified, but the number of people in analyzing one case is about 5 or 6. It's not one single person doing the job. It is 5 or 6 people. They do the job together and by and large they process a case within about half a day or 1 day depending upon the seriousness of the case and how big is the case. Now I'll give you an example of how difficult to understand and why this varied timeline is. There is a drug from AbbVie which was called as Abbott before. It's a very simple drug for Parkinson's. It's dopamine basically. But dopamine is a very old drug. So we were asked, would you do the case processing? We very gladly said yes, because the number of cases were very high. Or in suppose 10 lakhs a year or something was the case. We will do it very high. So he says, okay, how many people will you require? How much time will you require? So all the people did a calculation and told. What somebody forgot, we caught it before we submitted the details. What we forgot was the dopamine was given through an infusion pump. The infusion pump would pump it into a gastrostomy, that is, create a hole in the abdomen in the stomach, and the hole is outside. So it goes through that. And that is how the drug was given. Now you can have an adverse event because of the drug. You can have an adverse event because of the pump malfunctioning. You can have an adverse event because of the gastrostomy which is done. So to process a simple case, it takes about 1.5 days. Normally you would have taken hardly about half an hour or 1 hour. But it takes 1.5 days. So all your calculations can go wrong of what you can do. So you need to be very careful when you analyze it as to how is the drug given, what are the other things which are associated with the drug, and then say I can process it in this way or that way. Next one. Now who regulates it? In the US it is CDSCO. There is a PVP council in India which is there, Pharmacovigilance Council. They have created PV centers in different medical college hospitals in India. It started with 5 or 6 colleges. Now that is very common. You can all report cases there also, or you can report it directly to CDSCO. In Japan, it is the Ministry of Health and Labour Welfare where it is reported. EU is the EMA. UK is MHRA and US is the FDA. These are the most common ones. You also have to report sometimes to individual countries in Africa or Latin America or specifically in Asia. But that rules are there which once you get more into pharmacovigilance, you will understand those rules. Now this is how a case is processed. Now, as I said, when you have a collection of cases, you need to analyze the data in its totality. you get one case, you can't say that the incidence of adverse event is that. You need to have a collective thing. And there, if the drug is very new in the market, you need to submit these aggregate reports every 6 months. So collect the 6 months data, analyze it, tell how serious, non-serious split is, which patient population it has occurred. There is a way to write an aggregate report, and it gives any doctor or a regulator at one glance, okay, when we launched the drug, the status was this. Now today, after being used for 6 months, the status is this. It is bad or good can be assessed on that basis of that aggregate report. As the drug gets older, you need to submit it every year. As it gets more older, you submit it after 2 years. After that, individual regulatory bodies decide whether you need to further submit or you need to stop. So that is how aggregate reports are done. It is done to analyze the risk benefit of a drug over a period of time. Next one. Writing of an aggregate report takes about 6 months. It's not an easy task to write an aggregate report, because in the 6 months you may have thousands of reactions and you will have to study all the reactions individually. Now in US, before the drug is approved, in US you have to submit an aggregate report which is known as the IND annual report. IND is investigational new drug. So you need to collectively report that. In EU, the same thing is called an ASR, or an annual safety report. Today the name has been changed to known as Development Safety Update Report. Once the drug is in the market, you submit post-approval safety reports, which are known as PSURs, PADRs or PEBRs, depending upon which country it is and who has taken. So the name nomenclature has changed over a period of time. The content is by and large the same. As I said, this is done to enhance the risk-benefit analysis and to give you confidence whether the adverse event which you saw in the clinical trials have increased, decreased, new have come in, or what. So this aggregate reports give you that kind of an understanding. Next one. Now after you are submitting this adverse events and you keep getting new terminology, and you keep getting new adverse events over a time, what you will see is that suddenly you have a reaction which you have not seen. That suddenly some number of deaths have increased or number of hospitalizations have increased. Now this particular change in a pattern of adverse event which occurs is known as a signal. Signal means it is an indication to you that something is happening and you need to investigate it. Next one. These are actual definitions, so you see the slide, it is a horrible slide, but the definition is written there, so anybody is interested, they then go to WHO and they will get all this information there. So you understand what is written there and don't read it, I'll tell you what exactly it is. So when you get a sudden burst of some reaction which you have either not seen or has suddenly increased in number, that is called, that event which occurred is called a signal. Now signal need not necessarily originate from the drug. It can occur from the disease per se itself. It can occur from the other concomitant medications which are taken, because they may act with each other and give you a serious adverse event, suddenly increase in number. Or it can occur in only a particular country, in particular ethnic population, it can also occur. So you are supposed to investigate it. And that is what is signal detection. So when you are doing drug safety, there is a separate team of signal detection which looks at all the adverse events which you have processed, and it looks for signals कि कुछ अच्छा बुरा क्या हो रहा है drug. And if they identify it, next one. The signals are done to protect the patient, discover adverse events, understand risk benefit and avoid catastrophic change. If you don't do signal detection, unless there are 10,000 deaths, you will not wake up. But if the signal detection occurs, you may wake up after 100 deaths only. So that is the benefit of signal detection. Next one. Now once the signal is detected, you need to prove that the signal is positive. Means it has occurred because of the drug. And there are some statistical models which are used, which give you a confirmation in a span of 6 months or 8 months or 1 year that the signal is positive or negative. And if the signal is positive, then you have to add it to the label. What is a label? In India, the world over, there are two things which are existing. One is known as the core data sheet, CDS. Core data sheet is usually in US. If you come to Europe, it is known as Summary of Product Characteristics or SMPC. This is the master copy of what the drug is. Its toxicology, its teratogenicity, its adverse event, dosage, formulation, structure, everything is in these two sheets. And from these sheets, you derive a label. What is a label? A label is an information which goes onto the strip, package, ampule, box, vial, packaging insert, abridged prescribing information, patient leaflets. These are all called labels. So from these two, you actually derive all of them. Now if your signal is positive, as one case, if your manufacturing has changed and excipients have changed, if your indications have been added, if adverse events need to be added, contraindications need to be added, if you have changed the vial to a different vial, any change which you do in the product or the carrier of the product, then you need to go to the regulators and seek an approval. So when an adverse event or a signal is confirmed positive, you change the sheet, you go to the regulator and say that this signal is positive, we are going to add this into it. And once it is added, you have to change all the labels wherever the adverse event has been reported. So like on an ampule, you usually have some adverse event at times, at some you don't have. But a package insert, you will always have that. So you change that. And the importance of signal detection and pharmacovigilance, it ends at a change or no change of a label. And hence, this is the last end at which the pharmacovigilance utility ends. It actually ends at positive or a negative signal. Then they hand it over to the medical department, who then brings about the change, hands it over to regulatory, and then they submit and take an approval. So this is the entire chain of events which occur in pharmacovigilance. Next one. Now once you have the drug into the market, this is something which is constantly being evaluated by an organization. So you collect the data, you identify, analyze the risk. If it is risk, you take precautionary measures. I'll give you certain examples of precautionary measures which are taken. Like if you take for example ranitidine only, it's an extreme case. But if it was restricted to being hygroscopic, your B complex is very hygroscopic, extremely hygroscopic. Now if you have a package which leaks air into the pack, you will find a capsule completely smashed. If you complain, the manufacturer will have to look at it and change the packaging. Or if you see a vial suddenly changing the color, the content of it, the vial may be interacting with the drug. So you'll need to change the vial of the drug. So those are the kinds of things which you analyze and you quantify the risk. You measure it with benefit, because all drugs will have a problem. So is it more beneficial to give or it is bad to give? Depending upon that, you evaluate. You evaluate the risk benefit and then you minimize. So if you know it in advance, you withdraw the drug or change the formulation, change the packaging, change the vial, whatever you want to do. Or if you find that no, nothing of this will work, you go to the regulator and tell him, we have found this, we would like to withdraw the drug. Or the regulator finds it out. 99.99% regulator tells you, please withdraw the drug, enough of your nonsense. Pharma companies very rarely go to the regulator and say, mere ko drug withdraw karna. So that is what ends and benefits of pharmacovigilance is. Next one. Questions? Any questions or yeah?
As you said that we do drug testing on animals first.
Yes.
So how do we decide the dosage for humans? Like we do testing on humans because it is ethically wrong to test on humans directly. Because we have completely different anatomy and weight and mass, all that stuff.
Good question. Animals which we choose for studies predominantly is guinea pigs, rats, mice, and very rarely rhesus monkeys. Rarely. Now it has virtually stopped. What we try to do is we find the LD50 dose. What is the maximum dose which can kill? Lethal dose. LD50 is lethal dose. So we first find the lethal dose in any of this animal. Once you have found the lethal dose, you create an animal model of the disease. So you will say, for example, you are testing it on for diabetes. So I'll actually create a diabetic mouse, living diabetic mouse. You will destroy the pancreas, and the mouse will become diabetic, or the guinea pig will become diabetic. Now you will test an LD50 dose on it. It will say it gives you 100 units of insulin, it's toxic. Now you will reduce the dose then, and you will come to a level where it does not cause harm to the animal. Once you have got this, you do what is known as PK/PD modeling. I have not talked about it because it's very high science, but understand this much, that PK/PD modeling is done using softwares and computers, where you feed the animal toxicology data into it, all this LD50 dose, which animal, what you did, which disease, you feed it. And then models come out of it, statistical models come out which say that X dose in humans of this type should be sufficient to give you a therapeutic effect. So what usually they do is they go a step lower. So if it comes, say, use 2 milligram 3 times a day, so they will go to 1.5 or 1 milligram 3 times a day and test it in phase 1. Because phase 1 is to see what the drug does to the body and what the body does to the drug. It's done in a controlled environment within the hospital, so the patient lives in the hospital for 15-20 days. Every 30 minutes his blood sample is taken to understand how the fluctuation is occurring. His blood parameters and vital signs are monitored. So heart is monitored, pulse is monitored, respiratory rate is monitored. So various vital parameters are monitored. And if things go well, then they decide that X would be the dose. This is all andaj. And that andaj comes from experience, and the andaj comes from that modeling. Once you have come, okay, 1.5 was less, we should give 2. Then you will start with 2 milligram 3 times a day in phase 2.
As you said that we do drug testing on animals first.
Yes.
So how do we decide the dosage for humans? Like we do testing on humans because it is ethically wrong to test on humans directly. Because we have completely different anatomy and weight and mass, all that stuff.
Good question. Animals which we choose for studies predominantly is guinea pigs, rats, mice, and very rarely rhesus monkeys. Rarely. Now it has virtually stopped. What we try to do is we find the LD50 dose. What is the maximum dose which can kill? Lethal dose. LD50 is lethal dose. So we first find the lethal dose in any of this animal. Once you have found the lethal dose, you create an animal model of the disease. So you will say, for example, you are testing it on for diabetes. So I'll actually create a diabetic mouse, living diabetic mouse. You will destroy the pancreas, and the mouse will become diabetic, or the guinea pig will become diabetic. Now you will test an LD50 dose on it. It will say it gives you 100 units of insulin, it's toxic. Now you will reduce the dose then, and you will come to a level where it does not cause harm to the animal. Once you have got this, you do what is known as PK/PD modeling. I have not talked about it because it's very high science, but understand this much, that PK/PD modeling is done using softwares and computers, where you feed the animal toxicology data into it, all this LD50 dose, which animal, what you did, which disease, you feed it. And then models come out of it, statistical models come out which say that X dose in humans of this type should be sufficient to give you a therapeutic effect. So what usually they do is they go a step lower. So if it comes, say, use 2 milligram 3 times a day, so they will go to 1.5 or 1 milligram 3 times a day and test it in phase 1. Because phase 1 is to see what the drug does to the body and what the body does to the drug. It's done in a controlled environment within the hospital, so the patient lives in the hospital for 15-20 days. Every 30 minutes his blood sample is taken to understand how the fluctuation is occurring. His blood parameters and vital signs are monitored. So heart is monitored, pulse is monitored, respiratory rate is monitored. So various vital parameters are monitored. And if things go well, then they decide that X would be the dose. This is all andaj. And that andaj comes from experience and the andaj comes from that modeling. Once you have come, okay, 1.5 was less, we should give 2. Then you will start with 2 milligram 3 times a day in phase 2 and see very closely whether it is causing adverse events. So normally if they are suspicious, what they do is they will start with 1, 1.5, 2 and 2.5, all the 4 doses in similar kind of patients and see what the impact is. And wherever the benefit is maximum and the adverse event is minimum, they will fix that dose, or they may fix 2 doses. And then those 2 doses, they will test it in phase 3. So that is how they reach a dose. And that is why if you see, a dose is always in a range. Dose is not specific. If you say paracetamol, they will say 500 milligram to 1 gram at one time. So if you have a plain headache, take 500. Pharma companies have created 650, which is wrong. They have their own reasons why they have created it. But either it is 500 or it is 1000 milligram and multiples of it. So that is how the dose is given. It is predominantly done using the animal modeling studies and PK/PD data on a software. It's a whole branch of science which is known as clinical pharmacology, a part of clinical pharmacology. But they usually take PhD students of pharmacy in that, or they take clinical pharmacologist doctors, MD pharmacology doctors. So those 2 people are only preferred there. Statisticians help them.
Thank you, sir.
Yeah. Awaj.
Sir, what was the difference between human PV and animal PV?
Difference between
Human pharmacovigilance and animal pharmacovigilance.
what occurs, if you're talking of veterinary medicine, I won't be able to tell you. I'll tell you it in context of human drug development. When adverse events occur in animal models, that, as I said before, becomes a part of animal toxicology studies. So it's not called pharmacovigilance, it's called toxicology. Because it is analyzed in a different way, which a toxicologist does. When the same reactions occur in humans and you study them, it is known as drug safety or pharmacovigilance. So the reactions could be different, sensitivity could be different, severity could range, and hence 2 different individuals do that job. Two, 3 things can occur if the drug is given and if it is adverse effects which are adverse events which are occurring which are known and not very dangerous. Take for example, you have taken sorbitrate and it gives you headache. Sorbitrate is sublingual. So the moment a dose goes a little high of what you goes through the mucous membrane, you start having a headache. So you spit it out. You don't continue to take it. That is what is advised even in the prescribing information. So if such incidences are there, you don't do anything to the drug. But add a point in prescribing information that if you take this and have a headache, spit out the tablet. But this is for one which you are keeping it in the mouth. Suppose you have taken orally, and if, take for example, aspirin and you have severe regurgitation and vomiting and nausea, then the first thing to do is to stop the medication. Rarely and unfortunately in some patients you may have gastric ulceration occurred already when this occurs. Because all other painkillers are also similar. They may be here and there a difference. A little bit. Now I'll give you an example if you're talking of that. Aspirin gives rise to those problems, right? It's known. What is the alternative? Ibuprofen. Ibuprofen causes more problems, which is they came to know. Then what did they do? Take a COX-2 inhibitors. COX-2 inhibitors, then they found that it doesn't have this problem or very low, but it can give to cardiac problems. Now there is no painkiller. What do you do? So what you do is like an aspirin. What they say, खाने के बाद लेना और खाते समय लेना. Take an antacid along with you. So it minimizes the impact, but has the beneficial impact of pain killing or inflammation. Now, COX-2 inhibitors, you can't prevent a cardiac action. So you have to withdraw the drug. And that is how the drug was withdrawn. So that is the criteria on which you decide whether you want to continue. Oral hypoglycemic agents known to cause hypoglycemia. What do you do? Insulin will also cause hypoglycemia. Now what do you do? Sugar ka packet jeb mein leke ghumo. And make sure that your meals are proper, then the incidence is low. So it depends. That is what, as I last said, is the risk and benefit, which is overweighing. If the risk is overweighed, withdraw the drug or change the formulation if it is conducive, or withdraw the drug or continue the drug with certain precautions. Give him the mic. Sorry?
Thalidomide mainly used for?
Today. Thalidomide was mainly used as a tranquilizer first, then it is started using for nausea vomiting before it was withdrawn. Today, it's still there in the market in some places and they are trying to use it for HIV infection. But there is no evidence as of now. So some pockets may have thalidomide, but by and large it is not in the market and used for any therapeutic use. It has a sedative property, but there are better sedatives than thalidomide. So why do you give it? Now, if I tell you very simply, the biggest limitation of pharmacovigilance is how many people report the reactions. As I said, only 10% report. Of the total adverse event which actually occurs, only 10% are reported. So whatever you are evaluating is only the 10%. But because no data comes from anywhere else, I cannot assume that other data which was there. So I have 10%, in 10% मेरे पास 5% reactions positive आए हैं. अब 90% ने report नहीं किया तो 90 में से 45% will also be positive. I can't make that assumption. So I just leave that sample. So that is first is that a limitation. Okay, I am analyzing only a small sample of what actually occurs in the world. First restriction. The second is when you are analyzing a report, a collection of reports, for aggregate reporting, it's very different medical analysis than a single case analysis. It's very different. And interpretations can change dramatically. Hence, when aggregate reports interpretations change, it gets reflected into how you will analyze the future single case reports. So that is another limitation, that aggregate reports gives you a limited vision of the reported events. Aggregate reports gives you a vision which is different from the original thought process. So those are certain limitations. And these are scientific limitations. There are writing limitations, which as you start writing aggregate reports would be more conducive. English, for an example, English is a problem. The way you write a sentence is a problem. There is a specific way of writing it. The way you construct it is a problem. So all those things are practical writing problems, but not scientific problems. How do you overcome what? The first two scientific ones, if you have an adverse reaction tomorrow, start reporting. That will be the first step. Ask your physician to report it. But that is the only way you can do it. And of this 10% also, remember, US reports the maximum. So within this 10%, about 6% come from US. About 2 to 3% come from US. About another 1 or 2% comes from Japan. Rest of the world gives just 1%. But it has a population which is 60% of the world. So we are in a flawed state, but now we can't compel everybody to write. Your clinical research or any form of research is also in a bad shape today. I'm talking something else now. And the reason is we don't report failed experiments. We have stopped reporting failed experiments. Journals have stopped accepting failed experiments. But they fail to understand that a failure of an experiment, any experiment, is a stepping stone to a success or a safety of the next experiment which you are doing. And initially experiments were done like that. But today nobody wants to report it, whether the experimenter or the company or the college or the anybody, they don't want to report. So you are poorer and hence you see so many problems. That's the actual state of the world. Yeah.
Sir, some detailed about robotic surgery and who invented this.
Anybody else is interested, I can talk. If not, we can talk one-on-one. Because robotic surgery is something which is very deep and very extensive. And is everybody, rest of them is okay, because I would not like to waste other people's time. He is interested and I'll answer your question definitely. Others are interested, then only I'll speak. Robotic surgery is a little, keep it for a minute separate. Different robots which are used in medicine do different work. They're developed by multiple people and multiple companies, not one company. The basic principle is same as of a computer. I'm simplifying it. Don't take me at the word. I'm simplifying to explain. When you have a computer and you switch it on and you click on the windows, it opens windows. Then if you want Microsoft Excel, you want PowerPoint, you want Word, you click on that and it opens. That is how the computer accepts a command and it opens what you want. Now, in a robotic surgery, the robo has different arms. उसके 20-25 हाथ हो सकते हैं, 2 हाथ हो सकते हैं, 1 हाथ हो सकता है, depending upon how complex you are doing. Each arm has a specific function. So one function can be cutting of the skin, one function can be cauterizing the blood, क्योंकि bleeding होता है तो bleeding stop करना पड़ता है, तो use an electric cautery and burn off the portion. दूसरे का है कि भाई अंदर जाके अपेंडिक्स करना है, अंदर जाके अपेंडिक्स को ढूंढा और निकाल के बाहर आया. Now all these instructions are given by the surgeon sitting outside the operation theater or within the operation theater. He has a screen and there is a camera lens in a probe in an arm which goes deep in and shows you all what the structure is. So what the general surgeon may visually see it when he is operating physically and personally, the same image he gets it on a TV screen and sees it in a more broad manner. He gives the instruction to different arms to do different things, and that is how he completes the surgery. It's a notion today that robotic surgery is painless and robotic surgery is safe and robotic surgery is quick. All this is notion, it's marketing gimmicks. Because ultimately it is the skill of the surgeon which will decide whether the surgery is successful or not the robo. Normally क्या रोबो को आप ओटी में छोड़ दोगे और उसको एक इंस्ट्रक्शन दोगे कर डाल एक लेप्रोटोमी, तो वो जाके लेप्रोटोमी नहीं कर सकता वो. Somebody has to continuously instruct it. Now they will say AI/ML आ गया. AI/ML is one of the most dangerous things in the world if not used properly. That I will talk when I talk on AM. So various robots have been developed for various types of surgery. So brain surgery के लिए एक है, abdominal surgery के लिए एक है, heart surgery के लिए एक है, kidney replacement के लिए अलग है, prostate के लिए अलग है. So अलग अलग robots are developed, but not all robots are as good. Today also they have huge problems because at the end of the day they are electronic equipments. आपके इसमें भी वायरस घुस जाता है कंप्यूटर में तो सब खराब हो जाता है. और suddenly you have seen virus, without a virus the computer just shuts down. So these are called bugs. So those bugs are there in the program and that can occur. Yeah.
So what are the different risk minimizing strategies and how they are being implemented in pharmacovigilance?
Reducing risk strategies for what? Which risk?
While testing the drugs.
While testing the drugs. The first risk mitigation strategy which they use is doing animal studies. Because if there are many drugs which fail. If you are testing 100 drugs, just one drug reaches the human studies. Rest all of them fail. So that is the first preventive mechanism. Now today, because the animal testing is not done, is done at an infrequent pace, they have created digital twins. Digital twin is a digital image of an organ or of an animal or of a human body. It's called as digital twin because it is similar, not same. It's similar or very close to how a human body or an animal will behave. So they do these experiments on the visual twin, virtual twin, and try to minimize it. But no matter what process you use and what you do and what technology you use, you cannot prevent clinical trials in humans, which has some or the other risk always there. You can only minimize it by these techniques. You cannot abolish the risk. So you always be very thankful to those patients who have subjected themselves to phase 1, 2 and 3 trial. भगवान के सामने हाथ जोड़ते हैं, उन पेशेंट्स के सामने भी हाथ जोड़ो, because if they would not have subjected themselves, you would not have seen a drug in the market. It's like to medicine it is said that the body in which you study anatomy, the dead body, make sure you give maximum respect to it. And once you start learning, respecting that body, you will learn better. There is actually, it is taught like that in medicine. I don't know what's happening today, but we were taught definitely like that. So these are certain very sacred things beyond science which you need to do, but you can only minimize risk. You cannot abolish it. Yeah. You asked me a very advanced question. Today we have multiple branches of clinical research, right? But no branch is independent of the other. People have a very wrong notion that I am a specialist of data management, so I don't need to understand drug safety. Or I am a specialist of clinical operations. मेरे को बाकी से क्या मतलब है? Fine, if you are contented with that, then you remain that. But if you want to understand the entire process of clinical development till the drug is withdrawn, then all the things, that is clinical operations, medical writing, regulatory affairs, drug safety, data management, biostatistics, they are all linked to each other. Because one hands over the data to the next. If he does something wrong, the future also will be wrong. And there is continuous feedback going on from data management to drug safety and vice versa, to medical writing, to evaluators. There is a continuous stream of information which flows. So if you really want to be a clinical research guy, you cannot say I am an expert in clinical operations or drug safety or CDM. You have to learn each one of them, or at least you have to learn one as major and be really a experienced person is there. You cannot be an expert there also. You can be a good experienced person there. And all the other paraphernalia, as you raise the question, you should know the connections between each one of them. Data management, you do the basic data entry, which is clinical and patient data. You also do safety data entry. But the analysis you do it is in this thing. So when you reconcile the data, the two get connected. When you write a report, the biostatistics guy gets connected, the medical writing guy gets connected. And he can have many questions which he will ask the safety guy to clarify or the data management guy to clarify. So like that they are connected. Last, it comes to regulatory. When the regulatory submits a report to the regulatory agencies, they have a lot of medical questions, manufacturing questions, quality control of the drug questions, excipient questions. All that has to be answered by individual departments, but they go through regulatory. That does not mean regulatory is an expert. But regulatory has an experience in how to deal and what questions come and how to answer them. So they will guide you. So that way all the chain is connected with each other But very unfortunately today people have isolated them and say ki no. So it's like you go to a hand surgeon, so he is a specialist of hand, he doesn't know whether it is right mein ye hai to left mein kya ho raha hai. And doctors or clinical researchers cannot be like that. They cannot be specialists, they have to be generalists, understand all, and that is the true essence of clinical research, not a specialist in something. But as you say, as I said, unfortunately, people have segregated themselves like that, so they don't cross domains. I have crossed domains, only reason being, when I first entered clinical research, I was supposed to write the protocol, do the data management, data entry, drug safety, evaluation, prepare a report and submit. So we and my age people were compelled to do everything, not that out of choice we did it. हमारे पास बहुत बड़ा brain था इसलिए हमने नहीं किया, हमको कोई चारा नहीं था, तो हमको सब कुछ करना पड़ा. But then we realized that if you don't do, you will not understand the whole of drug development, and hence you need to do it. So when you start a career, think on those lines. Don't think that I want to be. If you're thinking and you're happy with that, then stick to it. But if you want the overall understanding, then you will have to go through the route which I am talking.
Statistics research, that how do we take the sample size? What is the criteria to take the sample size in clinical trials?
Hello.
You have asked a question for which I can take a 5-day session. In a very short manner, if I tell you, sample size is decided on multiple criteria. A couple of important, and another thing which you should understand is a statistical significance which you ultimately prove does not mean that it is clinically important. Right? Do you know the difference between the two? Statistical significance of less than 0.05 is what is normally said is statistically significant. 95% confident interval, and you are sure. Significance in statistics means that if you repeat the experiment, you are likely to get similar results. 95% confidence means 95% you will get that, 5% you may or may not get. Where does it say whether the result was good, bad or ugly? So it is just a result. If you want to clinically interpret it, then statistical significance is not the criteria. Right? But why do you use then statistical significance? Is to ensure that if the experiment was repeated and if it was clinically significant, at least you would have derived a proper value. Unfortunately, today, people have understood statistical significance equivalent to clinical significance, which is wrong. So when you calculate a sample size, you first decide what significance you want. So you can say I want 0.05 or I want 95% confidence interval, 99% confidence interval, all that you talk. Then you say, suppose I am trying to experiment with an antihypertensive drug. So a physician would say that I want the blood pressure to drop from 170 diastolic or 100 diastolic or whatever by 20 millimeters of mercury. Diastolic and systolic, I want from what the patient had, 20 millimeters of mercury, I want both the drops. Or he can say 20 here, 10 here, whatever. Now they will consider that as a value. The end point is that it has to come like this. Then they will say that to achieve this with a statistical significance, they will have formulas for that. Then they will say that I want it a multivariate kind of a population. So I may do it in India, I may do it in US, I may do it in UK, Germany. So they will consider that. Then they will consider approximately, and this is done on the basis of epidemiology studies, in a given population, what is the prevalence and incidence of the disease. So you may say that the disease is prevalent in every 1 in 100,000 patients. So you will take that into consideration. Like this, multiple parameters are taken, endpoints are taken, age is taken into consideration, and other factors before they calculate a sample size. It's not an easy process. It's not 100% accurate process, but it gives you some amount of semblance that what you are doing may turn out correct. The biggest problem which we have is not in sample, but in interpretation of the result, whether it is clinically significant or not. The second is when you were initially calculating statistical samples, you are dealing with causes of a disease. Today you are dealing with risk factor of diseases. And very unfortunately, risk and cause has become synonymous with each other. You go to any cardiologist and ask, is cholesterol a risk or a cause? So he'll say it is a cause. Very unfortunately, cholesterol is a risk along with 5 other risk factors of cardiac problems. Now how much it is a risk? Nobody knows because studies are not done. Now how do you calculate that into your sample size? So general, cholesterol is a problem, consider it. वहीं आपका पहले sample wrong होना शुरू होता है. So these are pitfalls and benefits of a sample size. But today, in a lack of anything, we take that. Your point less than 0.5 is an arbitrary figure. It's not a scientifically derived figure. Fisher thought that 1 in 20 is good. So Fisher said, and Fisher was a great guy at that time. अब हर आदमी जो great हो कुछ भी बोल दे उसका ये मतलब थोड़ी कि वो सही है. So that 1 in 20 is continuing till today. But even that time it was wrong. There was no scientific validation of it. He just threw out a number from his head. So everybody accepted it. Who will question Fisher? They accepted it. He himself has written that I am not sure of that number. So that is how, so statistics, there are 3 types of lies as they say. Lies, white lies and statistics. Choose one of them.
Hello. Thank you, sir, for your wise words. Today marks the end of the 3rd day of our workshop. We feel gratitude towards Dr. Shirish Sherlekar, sir, for enlightening us with his practical expertise and experience, in-depth knowledge, and valuable insights on the topic of pharmacovigilance and drug safety. We are thankful for your time, effort, clarity with which you explained the concepts. We are truly grateful to all of you for making this day successful, and we look forward to welcome you to the next session. Tomorrow's session will start at 10:30 when you will be the same SOET hall. Thank you. There are high tea and refreshment arrangements present at the designated room. Thank you.